in the field when we were completing some of the field work. So let us get started. So this is just an overview of what we'll be covering tonight. We're going to start with background information and objectives of the research, followed by a site description, then methodology and fieldwork, which you could see in this picture was very enjoyable, followed by some results, as well as conclusions and implications, as well as suggestions for future work. And finally, just acknowledgements, some big thank yous, and we're excited. So I wanted to start off with some broad definitions for those of us who might not be as familiar with the science behind my research project. Starting off, the big one is eutrophication, which is a process by which a body of water is enriched in dissolved nutrients that stimulate growth of aquatic plant life, resulting in a depletion of dissolved oxygen. And as you can see in this picture on the right, it forms a green covering on the water, which is also related to harmful algal blooms, otherwise known as HABs. This is the next definition, and this is a result from rapid growth of algae that can harm people, animals, or the local environment. So again, that's that green color that is can be due to the algal abundance. And further, there is also cyanobacterium. This is commonly known as blue-green algae. And this is a photosynthetic single-celled bacteria that forms filaments, sheets, or spheres, and can be found in diverse environments. And actually, this picture on the bottom right of the screen is a species of cyanobacterium that I found in a water sample from my project. It's seen in this circle pointing to, with the pointer from the microscope, it's kind of hard to see, but that's a species of cyanobacterium that we saw on the fall kill. And finally, something that I'll be referring to that might be unfamiliar to a few people is microsystems, which is a species of cyanobacterium. These are really common and they're harmful because they produce hepatotoxins, which can be harmful to the liver, and they also can be carcinogenic. Following up with some more definitions, chlorophyll A I'll refer to, and it's just a measure of the amount of algae in a body of water. It produces that green color. And you can see a little visual of it to the right here. A really important definition is macrophyte. This is the plant that grows in or near water and is visible to the naked eye. This is the basis of my research, and this is just one of the species that I found on my research, one of the lily pads. Another definition is denitrification, which is the reduction of nitrogen or nitrogen compounds, most commonly reduced by bacteria, and this results in the escape of nitrogen into the air into a usable form. And you can see this fun little graphic that I found as a bacteria, which is reducing nitrogen to a usable form. And finally, the last definition is watershed, which is an area of land from which water drains into a stream, river, lake, wetland, estuary, or another body of water. So, talking about eutrophication and all of these background information, I wanted to give a visual to give a clearer impression of what I'm talking about for those who might not be familiar. So this visual just goes over how eutrophication impacts the system over time. And you can see in the no problem and low impact zone, there's a lot of submerged aquatic vegetation. So from now on, I'll probably be referring to this as macrophytes, which is what my study is focused on. And as eutrophication symptoms increase over time, you can see that the macrophytes decrease along with it, as well as an increase in macroalgae, chlorophyll A, and nuisance or toxic blooms, which are also known as HABs. And in conjunction with this, there's a lot less dissolved oxygen in the water that's available for species that need it, such as those macrophytes. Because macrophytes overall, they have an inverse relationship with eutrophication. They're really good water quality indicators, and they're helpful for the health of a body of water overall because they produce a substrate for other organisms, as well as shelter for a lot of those bacteria that are helpful in denitrification. Yeah, and overall, they're just really good things to have in a body of water because, as you can see, as conditions worsen, they appear to decrease. And this is just a following image from that first one. I liked this one a lot because it shows how land usage can also impact a body of water in ways that we might not even think about. So the visuals are a bit small here, so I'll go into each one. They include industrialization in factories, urbanization, mostly residential, you can see with the houses there, agricultural, both farming on land and then animals, water runoff from rain or other weather events, and also wastewater treatment facilities input a lot of pollutants into water that can run off and then lead to problems downstream. And that's going to be really important in my research project, which I'll explain later on. But that's one of the major sources of nitrates and phosphorus, which you can see with these two little circles here. Introducing these two chemical compounds really affects the health of the body of water, as you can see in the worsening conditions. A lot of the influencing factors are present that were in the previous slide. So an X is through the macrophytes, but there's an abundance of macroalgae as well as chlorophyll A, and there's evidence of HABs. And it's important to recognize in this visual, the X's are also through humans using the bodies of water, either for recreation or fishing or even swimming. It's really not recommended that humans come into contact or even fish with bodies of water that are experiencing eutrophication, because like I mentioned slightly earlier with the definition of microsystems, they can be really harmful to our bodies as well as other animals that use these bodies of water as well. So it's just important. I think that that ties in really well to climate change and how warming surface temperatures make those cyanobacteria, the macroalgae, much more capable of dominating the surface and disallowing light to penetrate and feed those submerged aquatic vegetation. So in conjunction with climate change, it really is important to remember how the water will be impacted for us as well. And this will come up later on to tie it all together, but that's just some background information on eutrophic systems and eutrophication as a whole. And this information was really vital in forming my research question, which I'll get into now. And I feel like some of that information is helpful in understanding why I felt that this study was important along the fall kill. So some objectives of this study that Dr. Zine and I talked about this past summer. The first one is to relate nutrient concentrations to macrophyte species frequency and abundance at a fine scale. In my study, this refers to 50-meter reaches along the fall kill, which I'll visualize later. A second objective was to relate in-stream conditions, mostly through nutrients by proxy of nitrates, to the surrounding land use, which was seen in that previous visual, how the surrounding land to the fall kill has a huge impact on the water quality and the in-stream water conditions. And finally, just to understand macrophyte species' responses to a changing nutrient profile. And with that, I just wanted to show another visual of what different growth forms of macrophytes are, since I'll refer to this later on. Both A and B refer to submerged aquatic macrophytes. C refers to floating aquatic macrophytes, and D is an emergent aquatic macrophyte. And then moving on to my study, some of those objectives were helpful in forming some knowledge gaps, which I'm going to talk about and which I wanted to explore through the site locations that I chose. So we'll start on the right-hand side of the screen. This is a visual of the Hudson River watersheds that are present within the Hudson River Valley. And you can see through the black square is where Hyde Park is located, which is where this study took place. And these are located within the Hudson River Estuary watershed, which is Hyde Park and Poughkeepsie, where I'm at Marist. And then moving to the left side of the screen, you can see that I zoomed into, again, Hyde Park within Dutchess County, within New York State, but then the base map shows the stretch of fall kill that I actually studied. So each of the blue boxes represent a 50-meter So there's 50 meters between each one, and there are 42 of them in total. And this entire stretch of the Fallkill was 1.73 kilometers long. And it was of interest to me because, as you can see, the surrounding land to the Fallkill is a lot of hard surfaces, so impervious surfaces. It's actually 55% covered by impervious surfaces, which are strong rerouters of pollutants into the Fallkill. But the most important thing to realize in this visual is this circular area right here at the top of the stretch of the Fallkill in which I studied. This is the Hyde Park Trailer Park home. These trailer parks have individualized septic systems. So like I said earlier, wastewater was an important producer of nutrients that can be rerouted into streams. And you can see here just how close that these septic systems really are to the stream. So it's potential that the septic systems here were able to produce a form of nutrient loading from a point source that can be seen downstream. So this white arrow that you can see in this map is pointing downstream or southwest or to the lower left-hand corner of your screen, moving towards the Hudson River. And I'll explain implications of this when I share the nutrient profile, which I found. So next, I'm going to go over some methods that I employed in the field. The first one that I'll talk about is in situ water quality conditions. These were taken every 50 meters with a calibrated multi-probe that takes chemical readings. It's called a YSI probe. So I did this 2 days. It took like the entire day. It was a pretty long day wading through the Fallkill. So we covered each of the 42 sites with the chemical probe, taking readings of nitrate concentrations, dissolved oxygen, pH, temperature, as well as specific conductivity. And this was done in September to qualify the dry period or the lower flows with the warmer temperatures, as well as in November. And this qualified the wet period or the colder period with higher stream flows. And next, I took biometric observations. So this is where the macrophytes come into play. This took place throughout the fall semester, so between September and November. And these biometric observations followed the brown blanket sampling approach, which measured percent abundance of each species, which you can see in the picture here. There are a lot of species that fit in this 1 by 1 meter quadrat. So what this approach really aimed to do, it took transverse transects, which is basically a long tape measure at each of the 50-meter reaches. So within each 50-meter reach, we took a random quadrat sample every 5 meters. And this was randomized based on horizontal or vertical randomization tactics. And so each 50-meter reach had 10 quadrat samples of percent abundance of each species that were seen from an overhead view. So it was a lot of data that was taken to fully characterize this site, which was really helpful in the conclusions that I will list later on. And another thing that we did at the start of each 50-meter reach was take environmental conditions, basically just to further characterize each site. So this included the depth of the stream, light coverage from overhead based on shading from trees, as well as substrate class size from below. So the size of the pebbles or sand or silt. So again, this is just a table of the abiotic variables that we took in order to fully characterize the site. The water quality conditions were taken with the chemical multi-probe, the YSI probe. And like I said, these included nitrates, specific conductivity, pH, temperature, and dissolved oxygen. And the environmental conditions were taken with multiple instruments in the field, and they included depth, light availability, and substrate class size. And this big, large table that you don't have to read every name, this is just a list of the 50 species that I identified along this 1.73 kilometer stretch. I wanted to include this just to provide the information of all the data collected, because later on I'll discuss only 3 or 4 of these species in length. So this is just to provide the information that there were a lot more than that, and this table includes both the scientific name as well as the common name. And then after all the field work, I completed some statistical analyses. So I calculated the frequency of each species using the following equation. So the frequency of a species, which is signified by the variable I, was calculated by the number of quadrats that the species was present at, divided by the total number. So like I said, that was 10 per reach. So if a species was seen at 5 of the quadrats in 1 reach, it would be a 0.5 frequency rating. And I also calculated the abundance, which was completed through this equation. And this was a summation. So the variable S is significant of the percent coverage from 0 to 100 that was taken for a species, and for the amount of quadrats that it was present at. So it was divided by that number and then summed up for all the 42 sites, gave the overall abundance of the species. And these 2 equations were again taken from the brown blanket paper that I utilized the field methods for. And further, after calculating these values, I just found some significant relationships for both the wet and dry period nitrate concentrations in both R and Excel, and I'll explain what R is a bit more for those of us who are not familiar. And finally, I just completed a spatial analysis of the site in QGIS, which is a mapping platform. So this is a Pearson's correlation analysis. Again, this is a pretty overwhelming visual. This is what R is. It's a coding application. And what it does in the correlation analysis is that it takes every variable that was calculated. So the abiotic and biotic variables that I listed previously, it measures each one against the other to find significant relationships. So the Pearson's correlation analysis that I have for all of the variables probably wouldn't even fit on this slide. So this is just the found relationships that were significant to wet condition nitrates. So from that list, only 3 of the macrophyte species were found to be significant, and the remaining are abiotic variables that were found to be significant. So the outlines in red are significant of the most significant relationships. So for the wet nitrate conditions, it was mostly related to wet condition pH and wet condition specific conductivity. And this will be explained later on, but it's important to recognize that nitrate conditions are closely related to other conditions that could influence the presence or absence of a species. And this is the exact same thing just for the dry conditions. So everything else pertains. There were, again, only 3 biotic variables that were most significant to dry nitrate concentrations. And again, pH was found to be significantly related on a really small scale. So that's just important for covariance of abiotic variables influencing, again, like I said, the species presence or abundance along the fall kill. And this is a visual of a map that I created in QGIS. So this is the nutrient profile. On the left, you can see the dry conditions, and then on the right, you can see the wet conditions. And I like this visual because it shows that there is really big difference in nitrate concentrations between the 2 periods. So the dry conditions were taken in September and the wet conditions were taken in November. And with the colder temperatures, but the higher stream flow, you can see that nutrients were much more capable of moving around and settling, which is why the values were much higher. And then, like I mentioned, with the septic systems and the introduction of primarily of nitrates to the fall kill in such proximity, you can see that high values were seen downstream at this little indent. The highest values for both dry and wet conditions were seen just downstream of that. So that's just a little bit further evidence of some of the ways in which nutrients settle and are affected downstream of a point source pollutant. And all of these nutrient ratings are correlated to the biotic macrophyte species that were found at each of these 50-meter reaches. And now getting into some of the results. So these are going to be results between nitrates and biotic variables. So the first one that I'm going to talk about is Ludwig Polustrus. You can see it in the top right of your screen there. If you need a visual, it's also known as marsh seed box. This was a negative relationship. So as concentrations of nitrates increased, the abundance of this species appeared to decrease. The next biotic variable that was significantly related to dry condition nutrient periods was Persicaria Hydropiperoides. This is also known as swamp smartweed, and you can see it in the top right hand again of your screen. This is one of the only species that showed a positive relationship with increasing nitrate concentrations. So as the nitrate concentrations increased, the abundance of this species appeared to increase along with it. Now moving on to wet condition nitrate periods. So these are the higher nitrate concentration ratings. The first significantly related variable was floating aquatic macrophytes. So this shows a negative relationship, which actually doesn't corroborate with a lot of the scientific research that has been done in eutrophic waters. Usually eutrophication, eutrophied bodies of water have an abundance of floating macrophyte species that just litter the surface of the water. And this finding didn't align with those other research findings. So I'll explain this a bit later on, but some reasons for this could be the size of the floating macrophytes that I saw and the diffusion of these species. They were small and very able to be spread out. And in addition, the other sources that cited this relationship that eutrophication usually leads to an abundance of floating species, they were all completed in lake ecosystems. So it's just something to think about. I'll go into it more moving forward, but so this negative relationship is a pretty significant finding for this research project. Another variable was Spargadium Americanum. This also showed a negative relationship with nitrate concentrations, so its abundance appeared to decrease as nitrate concentrations increased. And finally, the Lemna species were also found to have a negative relationship with nitrate concentration. So this is duckweed. This is a floating macrophyte. This was probably the most abundant species that was seen. But like I said, it's really small. This visual doesn't really do its size justice, but it's about the size of a bean or like a peanut. It's really small and it spreads out a lot based on the flow of the water. So that could be a reason that this also showed a negative relationship, but this does align with the finding that floating species had negative relationships with increasing nitrate concentrations. And this is just kind of an overview table of the species that I stated previously. These are all the 5 species that were most significantly related to nitrate concentrations, as well as their common names and scientific names, some visuals, and the abundance and frequency of each one that was calculated through the equations that I listed previously. So moving on from results are the conclusions that I drew from these findings. The first is that there is a confluence of multiple controls on a species that can either reduce or amplify the effects of a single stressor. So that was kind of the purpose of the correlational matrices. They just show that multiple abiotic variables can work together because they're so significantly related at each site in order to influence the presence or the absence of a species. And in this paper that's cited here, the Geiswein paper, it was actually found that in this study, when there were multiple controls over a single stressor, the effects of the stressor were actually reduced based on the confluence of effects. However, I do believe that it would be helpful to study this in a more experimental way, which I'll go into further when I explain future suggestions for the study. But that's just something to keep in mind based on the statistical analyses of these findings. The other conclusion that I drew was that emergent aquatic macrophytes were the most abundant growth form in terms of percentage compared to floating. And like I said, this doesn't corroborate with other scientific findings in other eutrophied bodies of water. However, these other studies took place in either lake ecosystems or stream and river basin ecosystems, which were not in the US. So this finding could be novel for this particular ecosystem. So this was a low flow and pretty small scale stream. So it could be potential that this is the way in which lower flow and smaller scale streams respond to eutrophication. And finally, there were some very significant species specific relationships that were found that could be helpful in further bioremediation. So these species that you can see at the bottom of your screen here were found in other studies, the studies that are listed on the last bullet point. They were found as obligate wetland species to have very significant denitrification potentials in these similar studies and similar ecosystems. So each of these species was significantly related to nitrates, and they were found previously to have denitrification potentials, which could be really helpful moving forward for the Fallkill Watershed Coalition in order to implement some forms of bioremediation. Some further implications of these conclusions and findings. The first one lays in the found relationships that could be indicative of tolerant species. So those include swamp dogwood and swamp smartweed. These are the positive relationships with nitrate concentrations. But on the flip side, there was also an evident intolerable threshold between species and nitrate concentrations. So this includes marsh seed box, duckweed, and burrweed. And this just could be useful application in watershed management as well as bioremediation, but it is just really helpful information that that large species list that I listed at the beginning was whittled down to these few species that were found to be tolerant. Another implication to water quality is that species richness. So richness is the number of different species that are present at a site. The threshold of this parameter seem to have a drop off at approximately 3 milligrams per liter of nitrates. And then in the wet nitrate conditions where the values were higher, the species richness values seem to increase again. So this could be either indicative of increasing beds of tolerant species like the ones that were listed above, or it also could be indicative of creating an environment that's really helpful for non-native species or invasive species, because that was seen. A lot of the species identified in a particular stretch actually were non-native. So this could also add to the species richness, because although there weren't a lot of them, they did show different values of species that were present at this area. So that could just be another indication of eutrophication. And finally, this is something that I've been touching on again and again, but the community structural shifts in aquatic macrophyte growth forms and algal abundance could be suggestive of eutrophic conditions for this particular type of ecosystem. So these unappealing looking photos at the bottom are some photos that I took in the field of various algal species, mostly green filamentous algae, which is rampant along the Fallkill. And algae actually isn't considered a macrophyte, so it didn't go into any of the calculations for abundance or frequency. But based on my characterization of the stream, it was probably present at almost every single quadrat, which is saying something because there were a lot of quadrats. So this is a really important finding that could be indicative of eutrophic conditions, because the Department of Environmental Conservation actually doesn't define a numerical standard for nutrient concentrations that indicate eutrophic conditions. It actually just states that in abundance of algal growth, weeds, or slimes that impair the functionality of water can be indicative of eutrophication, which was clearly seen throughout my observations of the Fallkill. So that's just kind of another source of data that shows eutrophication is definitely trending in this area of the Fallkill. So I did want to apply these findings to other Hudson Valley watersheds. Although this one is very specific and specialized, I do think that there are a lot of sources out there, like the 2 links that are present on this slide, which will be again stated at the end, but there's more information for a lot of various creeks and streams that are present within the Hudson Valley watersheds. Because we rely on this water so heavily, as you can see through this pie chart, over 80% of the population is served through surface water. So that's the blue value here, based on community public water systems. And this is within the Hudson River estuary watershed, which is where I am in Poughkeepsie and where Hyde Park is. But there's also a chunk that is also served by groundwater. So all of these factors together with the visuals that I showed at the beginning, how activities on land can influence the water that we rely on for recreation, for drinking, for cooking, for brushing our teeth, going to the bathroom, showering, all of that stuff could affect our water quality and the way in which we can continue living our lives in the manner in which we do. So with that, I just wanted to offer some suggestions for future studies. The first of which is that there definitely needs to be a species-specific bioremediation practice within the Fallkill watershed. And I think that this could be really helpful if an experimental study was completed in order to find the mechanistic function of the species that I listed. So the marsh seed box, the swamp smartweed, and the burrweed in being potent denitrification sources in order to best implement bioremediation along the Fallkill. An experimental study would really be helpful in just finding out the ways in which these species of aquatic macrophytes utilize the nutrients and where they're most potently able to utilize the nutrients that are being loaded into the streams. And finally, I hope to publish this research in the Ecohydrology Journal just in order to elicit some more support and longevity for this study that can hopefully implement some experimental studies. And overall, I just hope that this can be helpful information in order for the Fallkill Watershed Coalition to introduce bioremediation as a potential combatant of eutrophication. So I'll just end off with some acknowledgements. I'd like to thank my peers, Natalie Kelchner, Emma Butzler, Luke Hatch, Samantha Roberts, Melina Sorreo, Jason Velez, and Kristen Van Allen for their dedicated help in the field. It was not easy work, and they offered a lot of their Saturdays to wade through the mucky Fallkill. So I really appreciate that. I really couldn't have collected any of this data without all of them. I'd also like to thank Nat and Aaron Flynn for collecting and sharing their stream discharge data. And this data helped me qualify the difference between wet conditions in November and dry conditions in September. I'd also like to thank Dr. Feldman, my professor here at Marist. He aided to help me identify a lot of the aquatic macrophyte species that I couldn't have done on my own. And I'd also just like to thank Marist College, the Environmental Science and Policy Department, for allowing me to use all of the equipment and resources, as well as providing funding this past summer, along with the Carey Institute and the Mid-Hudson Young Environmental Scientist Program, which allowed me to formulate my research in an environmental education setting. So I really appreciated that. It helped me to work through a lot of that background information and explain it to some local high school students. And finally, I would just really like to thank Dr. Zion, who you guys heard at the beginning. He has helped me through a lot of various tasks of this research. And I just really appreciate his dedication and help to mentoring me and allowing me to complete this research project successfully. And just some of the other scientific articles which were helpful in drawing my conclusions. And thank you everyone for listening. Listed are my email as well as Dr. Zion's email. And for more information, these are the links that I found would be helpful for more information, as well as ways to get involved. And the final link is the link to our lab group if you're interested in finding out some more information about that. But thank you everyone for listening. I really appreciate it. And I hope you enjoyed the talk.
Great, Katey, thank you so much for that presentation. We have time for some questions, and so if you have questions, again, please use the Q&A box to put those in. The first question, and I have a question too, if we can get to it. The first question actually came in at the beginning, but I'm going to ask it now. Does this presentation apply to phosphorus? So you focused a lot on nitrate in this presentation. I'm wondering if you can speak to what we might expect or think about related to phosphorus.
Yeah, that's a good question. Based on my research, before I even started this study, nitrates seem to be more indicative of eutrophication, especially because I wanted to focus on septic sewers. Nitrates were a big indicator of how that could be a pollutant. So I actually didn't measure phosphorus at all, although, like it said at the beginning, it is indicative of eutrophication as well, but I didn't focus on that just because of the specific parameters with which I wanted to investigate based on Hyde Park specifically.
Yeah, and I'll just add in as well, the difficulty in measuring phosphorus in situ, like in place in the stream, the way that we did all these other variables, it was very easy to measure nitrate with a electronic probe. That doesn't exist for an option for phosphorus in the same way. So in a lot of ways, the nitrate was just serving as a proxy for the types of different nutrient pollution you see in streams like the Fallkill throughout the region. So yeah, it kind of serves as a proxy there because it's something that we can actually easily measure quickly.
Great, thank you. So we've got a question in the chat, and I'll just remind folks, if you could use the Q&A rather than the chat to put your questions in, that helps us keep track. So we're not having so many boxes open here. But we've got a question from Richard from the Hyde Park CAC. Can you state your work in a few sentences that my reasonably intelligent grandparent would grasp immediately? And I'm wondering there, if I can paraphrase Richard's question, if he's going to report back to the Hyde Park Conservation Advisory Council, who are concerned about environmental conditions in the town of Hyde Park, what might be a couple of sentences that are the real message to bring to them?
I really like that question, actually. Going off of what I just said, I think I can whittle it down. So I think that the most important information is that there are 3 species that were found to be significantly related to nitrates. The most important of which I'm going to say is swamp smartweed, because it was found to be to have a positive relationship. So I think further study of these 3 or 4 different species and mechanistically how they can uptake nitrates or nutrients in general and utilize them in ways that will release them into the air into usable forms and not be harming the quality of the water. That, I think, is the most important thing moving forward. But just characterizing the stream, I think it was evident that there have been some inputs that have impaired the quality of the water, especially with the algal growths. So focusing on the point sources as well as the specific species that were found to be helpful in increasing nitrate concentrations, I think is really important moving forward.
Great, thank you. I'm going to ask my question, which was in your nutrient profile map, and then it showed all of the dots with different shades of red based on how high the nitrate concentration was. I noticed in the dry conditions map, there was one spot in particular that was a brighter red, and there were a couple other spots that were bright too. Wondering if you had any thoughts on what might be causing that, if you could speak to any observations. Obviously, this was not a track down kind of study, but if you had thoughts on that. And then also if you saw different macrophyte conditions there for places that are maybe more chronically exposed to heavy nitrate versus more of the, higher in dry periods, but higher in wet periods, but lower in dry periods.
Right. Yeah, I see what you're asking. Yeah, so Dr. Zine and I actually were talking about that yesterday, that one area.
Katey, can you go back to that slide so everyone can see those maps?
Sure. Might take a while. Yes, this one. So the one area here, I believe, is where you're talking about.
Yeah.
Yeah, so that is like I said, that is downstream of the Hyde Park trailer park home. And so the study was to see if conditions downstream of that point source were affected in terms of nitrate readings, which clearly they were, because at the same location in the wet conditions, it also showed some of the highest values. And if I am not mistaken, I believe here was one of the places where I saw an abundance of non-native species actually. So I am curious to further explore that relationship, if that is related, because like I said, species richness did increase with those increasing concentrations of nutrients, which could include the non-native species, as there were only a few of them, but a lot of different ones, which is what richness calculates. So yeah, I'm thinking that that area in general could be interesting based on the stream flow or the hydrology of the area, to see if for some reason that is harboring a lot of the nitrate concentrations from the point source.
Yeah. So Katie, can you take your mouse and put it on that little corner in the stream? Let's go to the, yeah, that's great. And then if you move it towards the left just slightly, you can see there's more lightly colored vegetation that kind of V's towards the north. And if you zoom in where that light color of vegetation, you can't do it now, but when you do zoom in, you can see that there's just less leaves on those trees. And they kind of point towards a point just to the northwest of that bend in the creek. Haven't had a chance to investigate that or figure out access to do that, but that whole region is kind of a wetland area, and it looks like there might be, say, a combined input from, you know, a septic system or a large septic system that might be designed in such a way that it doesn't have enough natural remediation going on in the subsurface, and then we're getting more nutrient rich effluent coming out kind of to that point just to the northwest of that bend, where all that kind of light colored vegetation all points to. So that's kind of the question that's out there, and that would be more of a management question that folks with, you know, Richard at the CAC and folks, you know, I'm part of the Fallkill Watershed Coalition. So something like this kind of helps us think about places we could further investigate that might have some potential issues on the landscape. And then I just want to build on for those. It was a good question. How do we sum up this for a grandparent or just anyone else that just wants to know really quick what we're doing? And I think if I want to say it in one sentence, you know, we're worried about harmful algal blooms downstream, and we're trying to figure out where those nutrients come from and if there's certain species. We found that swamp smartweed was a one that might actually be good at sucking up those nutrients that we could promote in terms of growth to get those nutrients out of the stream and maybe prevent those downstream issues. So I guess that wasn't exactly a sentence, but used a lot of semicolons maybe. Anyways, I'll hand it back to Emily for the next question.
Yeah, thanks, Zion. Sorry I cut you off before you got to answer that question.
Oh, no, that's fine.
What are the implications for biodiversity when using just 4 species for bioremediation?
That's a really good question. So I think the important part of bioremediation is that it's really tactical in where it's placed. So something like this that you see on your screens here, the profile, can show exactly where the bioremediative species would be most needed. So it's not like it's going to clear cut all the other species and remove them, because that would kind of be antithetical to what we're trying to promote here. But I think it's important to note specific locations in which they'd be most powerful and helpful in order to utilize the nitrates or the nutrients in general that are kind of congregating in certain areas.
Yeah, and a bioremediation effort wouldn't necessarily have to be distributed across the entire stream. There's impoundments. There's places where the water is pooled up throughout the fall kill, and we could have essentially a denitrification pond that was potentially had a certain species or two growing in it. That was a monoculture. It didn't influence hopefully the ecosystems upstream and downstream too much, but it gave us an opportunity to maybe grow things and then extract them periodically throughout the season to bring down those nutrient levels. So those are just some ideas. It's not really the expertise of myself or Katey with the work. We just wanted to help identify potential species in these watersheds in the Hudson Valley that might be related uniquely to nutrient levels.
Great. If phosphorus rather than nitrogen is the limiting nutrient in the fall kill, would macrophyte impacts on nitrate levels matter as much? Hard-hitting question from Ray Kepner.
Yeah, that is a hard one. I struggle with this question because there were sources in which I found stated that phosphorus was the limiting nutrient, and then there was also sources that stated that nitrates were the limiting nutrient. So I really don't know enough about it to give a successful answer. Maybe Dr. Zion could help with that one, but I did struggle with that because I did find conflicting sources on that exact topic.
Yeah. Yeah, I think there's many reasons for us to be concerned about phosphorus in the Hudson River watersheds, equally or even more so in some ways than nitrogen. Because of this study was focused on nitrate essentially as a proxy, and that's the only thing that we could relate to these vegetation species. But Katey analyzed the relationship to 50, close to 50 different species with all these water quality metrics, and only 3 and really only 1 was positively related to nitrate. If we had the phosphorus data at this high spatial resolution, which would be much more difficult to get than what we did here, we might see a lot of relationships to certain other species that are more controlled by that limitation in phosphorus. I would expect that, and from all the research, background research Katey's been doing, that's definitely the case that certain species are going to be more limited by phosphorus and other ones more by nitrogen. And we just happen to find a species that relates to nitrates because that's what we were able to do because of our methodological limitations. So a lot more options for future research on the phosphorus side.
Great, thank you. I see a very specific question. Besides lemna, what other floating species were present and part of the statistical analysis?
So like I showed at the very beginning, there were a lot of different types of lily pads that were floating species, and I would have to analyze my list again to go through the specific ones, but I know lemna, so duckweed, and there were various species of different lily pads that were present as well. Other than that, I'm trying to think. I don't remember off the top of my head, but I know those 2 were probably the most abundant floating species.
Great, thank you. In terms of the bioremediation, would encouraging certain species growth be a stream treatment possibility?
So yeah, I think that is kind of what bioremediation aims to do. It's not, like I said, it's not just monoculturing the entire riparian zone to be one species, but it's helpful to implement where you can. I think so. Along this stretch of the Falco particularly, it ran through a lot of people's backyards. So I feel like if it could be slowly but surely implemented into a few different zones just along this stream particularly, it could be really helpful.
Yeah, I think just like we have programs to help people plant certain riparian trees and other types of vegetation, I think helping identify aquatic species that might help in our environmental efforts is important, and that's what this study starts to try to do. And I think if the end goal is keeping all these nutrients from getting into the Hudson or into some of the lake impoundments that are along the fall kill, then you could focus remediation just upstream of those as well if you wanted to have focus sites. So you could have both approaches potentially as well.
Great. We've got time for one last question and then we're going to wrap up. Looking at these photos, the aerial photos of this section of Hyde Park, what is the wastewater treatment for the homes that are not the septic system? Do they have the trailer park septic system? Do they have their own septic systems? Are they on public wastewater that's served by a wastewater treatment plant? Do you know?
Usually homes don't have septic systems. If I'm not mistaken, they do public water to a wastewater treatment plant, I believe.
Well,
I know at my house at home, we don't have a septic system. We have
yeah.
like a storm drain. Is that what it is?
Yeah, so it's really variable. In this area, you going farther downstream, we hit sections that are connected to city sewer, but we do have a lot of individual septic systems in this section. Stay tuned for next year, because another student, Emma Butzler, who helped Katey with this, is looking at exactly that question. So she's mapping out each individual property and through the tax parcel data can see if they're on individual septic systems or in combined sewer systems. And then she has some grants out and hopefully get funded here to do a much more extensive look at the same nitrate data across a much longer section of the Fallkill and relate that to those different land uses and different approaches for wastewater treatment in homes up and down the Fallkill.
Great. Well, that's a perfect note to end on because we've got a strong foundation here. We're going to be looking forward to learning more about Emma's work and the next steps. I think it's really exciting and interesting to see this research coming out of, you know, the Fallkill and Marist and Hyde Park. Just looking at these aerial photos, you know, I think we can think about a lot of small streams in the Hudson Valley that look like this, that have this kind of development around them. So I think it makes for a really excellent case study. And of course, we're all concerned about nutrients and harmful algal blooms and the impact of climate change. So I just want to thank Katey again so much for your wonderful presentation and Zion for helping Katey in putting this all together. I neglected to thank Kate Meyerdirks, Dr. Kate Meyerdirks from Siena College, who heads up the Thirst Research Network. Kate has been here running technical support on the back end. So Kate, thank you so much for your help and partnership on this program. And we look forward to seeing you all next month for our next in the student research speaker series. So thanks again and have a great night.
Thanks, everyone.
Thanks, everyone.